Our guest today is Professor Diano Marrone from the Faculty of Science on our Waterloo campuses. Welcome, Diano.
Thank you.
Before we begin, I just remind you to please keep your microphones on mute. If you'd like to ask any questions, please do pop them in the chat. I will be monitoring the chat and I will try to get to them, time permitting. So if we could start you off, Diano, can you tell us a bit about your background? How did you end up here in Laurier? Is it a long and sordid story? Is that
Yes, as per usual. No, so I was born and raised in Winnipeg. Second generation, I suppose, Canadian. Both of my parents emigrated from Italy. I was the first person even in my extended family to go to university, so that was kind of a big deal for everyone. I went, as you can imagine, living in Winnipeg, to the University of Manitoba. Yeah. Did my bachelor's degree there. Went from there to master's and PhD in psychology and neuroscience at the University of Toronto. Mm-hmm. And then went off to a postdoctoral fellowship at the University of Arizona. As part of that, I also spent some time working at the California Regional Primate Center at UC Davis. Right. So I was doing some primate neuroscience research. And from there, I knew I had wanted to come back to Canada, was applying for jobs. Laurier, as luck would have it, happened to be advertising. I interviewed. This was the, I think, third interview that I had. They offered me the job. I was delighted to accept.
That's wonderful. Well, yeah. We're lucky to have you, Diano. Thanks for that background. Maybe you could describe for us a bit what your research program is. The broad general area that you're working in.
So I would describe the broad general area as neurobiology of memory. Mm-hmm. So in terms of memory function, how we learn new information, why we remember some things and not others, what determines what we forget and how quickly, all of those kinds of related questions have always, pretty much almost from the beginning of my academic career, been kind of the ones that I find the most intriguing, and so have been the focus of my research. Now, in terms of how you might break up that kind of great big umbrella question, that has actually changed a lot over the years as I've been an academic. I've had the luxury, currently, being a tenured full professor. You have the luxury to be able to explore and take a few more right angles, right, than you necessarily would earlier in your career. So my research program has changed, but these days, in that kind of umbrella question, I have two programs. One working with rodents, predominantly rats. So I have a rat model of diabetes, and this is kind of an early stages, what might be more equivalent to what people would be called metabolic syndrome, kind of very early stages of diabetes, and exploring some of the effects that kind of those early stages of diabetes have on the brain and on memory systems that might help, confer risk for things like dementia, because we know that diagnosis with diabetes increases risk of dementia diagnosis later in life. I didn't know that,
actually. That's interesting. So what is the kind of physiology, since diabetes, I'm no medical researcher, but I think of diabetes as related to your kidneys, not your brain.
But the brain is a very big, if I can word it this way, the brain is a very big sugar sink.
Right.
It's very energy hungry.
Yeah.
And so maybe based just on that, it's not surprising that it has effects. And, to really oversimplify what is probably a very long story, diabetes, it looks like from, our research and others, accelerates the aging process in the brain.
Huh. That's fascinating.
And so some of the physiological, even in the absence of pathology, some of the physiological changes that happen with age happen earlier in age and occur more rapidly in those who are diabetic, especially if blood sugar isn't controlled. right? And isn't controlled very early on.
What's the sort of time course of that degeneration? Is that something that happens rapidly? Does it happen to everybody that has diabetes?
That's, I mean, some of the physiological changes, yes. When you talk about relationship to dementia, that becomes a lot more complicated, right? Because dementia risk is very multifaceted, and a lot of people with a lot of risk factors don't ever develop any sort of dementia, and even those that do, it occurs decades later. So, you know, getting a tractable link between those two things is tricky. Yeah, but we know, you know, at the epidemiology level, kind of across populations, that, you know, the longer you have diabetes, and especially the longer it goes uncontrolled, the higher your risk of developing all kinds of dementia.
I could keep asking you questions about this for the next half hour, but I interrupted you. So you were saying you had two threads to your research program.
Oh, okay.
So you've got the diabetes model.
Yes, and I kind of went off on a tangent on that. The other side,
took on a tangent. So.
Fair enough. Tangents are always good. So the other side of that research program, which is going to at first sound very different, but it isn't, trust me, is I'm looking at the brains of birds and how their memory works, and especially how they navigate. Now, this is a great question, because it turns out in the brain, the same systems that are used for memory in general are used for navigation. And this is, you know, for instance, one of the reasons why, you know, one of the early stages, for instance, of dementia is, you know, loss of wayfinding. People get lost more often.
Okay, yeah.
It's because it's the same system that.
That's the hippocampus? Is that
the hippocampus, the temporal lobe cortex.
The only part of the brain I really know. I've really heard of. So I wanted to get it in there.
It's the best part. Yeah, so a lot of my research does revolve around the hippocampus, and the reason. So if we talk about the hippocampus for a second, the reason why I'm interested in birds, and this is where they're kind of two parts of or maybe two sides of the same coin.
Yeah,
I'm interested in the hippocampus of, you know, a rat, for instance, because it's just like ours. It has the same kinds of cells and the same number of connections and in the same pattern, you know, just scaled down a little bit, but everything is for the most part the same. On the flip side of that, I'm interested in the hippocampus of birds because it's really different.
Right.
So they have a structure that we call the hippocampus. It's still used for the same sorts of things in terms of memory and navigation, but it's accomplishing this in a much different way. And so I'm interested in how you know, nature has found a different solution to the same problem.
So connecting the two, like I get the you know, the question around the diabetes model is about sort of how you lose your memory through those physiological processes. In the birds, are you looking also at memory loss, or just in general how structure is related to function kind of thing?
I mean, you can look at memory loss, and people have. For the most part, my research has been about kind of basic memory function.
Okay.
But it's really, I mean, the two do go together and inform each other. I mean, if I can I give you an example?
Please, yeah.
Okay, so this is a project that I'm working on right now with actually Noah Miller, Laurier, who may very well be in the audience.
He is in the audience. I saw him. Yeah. And
Chelsea Damfus. So we are looking at birds. So one of the things that you can do, or one of the things the hippocampus, I would say, does, is that when you go to a specific place, certain cells in the hippocampus will light up, let's say become active. And it's not all of them, it's a small number, right? About a quarter.
Okay.
And when you go to a different, you know, place, to a different room, for instance, different cells will become active. So in room 1, cell 1 and 3 and 5 will be active. In room 2, cells 2 and 4 and 6. So you can, in essence, tell by the activity in the brain whether the brain, you know, believes, if I can word it that way, that you're in two different places. Okay, so it turns out that in mammals, you have two kinds of cells in the hippocampus, what's called pyramidal cells and granule cells. Now, most of the time they act pretty consistently, but if you create situations where there's a lot of interference, like for instance, exposing animals or even people to two different rooms that have a lot of the same cues, right? Same pictures on the wall, same chairs in the same location, etc. Where there's a lot of potential for interference, the two populations will separate their activity. Where a lot of the pyramidal
populations of cells,
where the pyramidal cells will say, these two rooms are the same, they couldn't possibly be more the same. And the granule cells will say, nope, these two places are different, they couldn't possibly be more different. And it's kind of the competition of the output from those cells is what determines where you believe you are.
Fascinating.
Yeah, so this is something that occurs in all mammals, and in part it's thought to occur because in the hippocampus you have a very peculiar arrangement with kind of a layer of these pyramidal cells, a layer of these granule cells that are connected in a very kind of specific way. And this anatomy is thought to determine this pattern of activity. That has been dogma for, I would say, 30 years, maybe more. And it turns out when you look at birds have the same populations of cells. Not quite, but they have something granule-like and something pyramidal-like.
Right.
They don't have this anatomical arrangement. Instead of being in these layers, it's kind of all jumbled together, as far as we know, kind of haphazardly. Yet, when you give birds this same kind of paradigm that you give to mammals, where you have these two events or rooms that are kind of the same, right? But there's a lot of potential for interference. It turns out those patterns in the birds do exactly the same thing. That the granule cells say, nope, these two places are different. And the pyramidal cells say, yep, these two places are the same. So something about our understanding of our understanding that the anatomy is required for these cells to do this is wrong. And so that's a situation where the kind of the bird research that I'm doing kind of informs things on the mammal side. That you have a different system to be able to test predictions about why things work the way they work.
This might be too hard a question, but how does the brain sort out the difference between those two sets of signals? If one set is telling you this is the same place and the other set is telling you it's not the same place.
Yeah, that's so.
Is it just one signal eventually overwhelms the other?
So are you talking about how the two become different, or how they kind of
how the brain, how your consciousness makes sense of those two different signals? Right, you're getting two signals, one from one set of cells, one from the other set, and they're telling you two different things. Does the brain just throw up its hands and go, I don't know, or does it preferentially pay attention to one of them, not the other? Or
so you have a whole separate system that does this kind of comparison, and also compares it to the sensory input that you're getting right now. So you can actually compare what these two other systems are retrieving, right, and compare that to current experience. And that, we don't always get it right.
Yeah, right. That's right. Yeah. Well, again, I'm sure we could carry on that part of the conversation for the rest of the time, but I have some other questions to ask you.
Sure.
What would you say has been your proudest moment as a researcher?
Oh boy, I have a lot of them. Of course, every time.
Maybe it doesn't have to be the proudest. You could just say a proud moment if you don't want to have to choose.
Fair enough. I was going to say every time you put out a paper and it finally gets accepted, it goes through that.
It's a little victory to celebrate.
Yeah, every single one of those is a proud moment. But no, there's a lot of proud moments. So I'm proud of a lot of things. Being able to get an academic position at a comprehensive university, proud moment. Getting tenure and promotion to full professor are all things I'm very proud of. Having students that have succeeded, completed, For instance, I've been fortunate enough that in my career thus far, I've supervised 3 postdoctoral fellows. All 3 have academic positions now, one of which has tenure. So this is, you can't help but feel proud of that from a, mentorship perspective of those moments. So it's a tough question to answer, in part because there's so many proud moments.
Yeah, absolutely. Yeah. One of the questions I've been asking guests on the show is around collaborations and whether you do them and don't do them and who your collaborators are. I know you mentioned Noam and Chelsea already, but are they your primary collaborators, or is this just a side project you started working on with them? Do you have other collaborators that you work closely with?
Yes, many.
I don't even have to ask, like some people, some guests I have to ask whether they collaborate at all, but since you're a scientist, I'm pretty sure you collaborate. That's the nature of science. So,
I mean, lots of, international collaborations all over the place. Right now, for instance, I'm co-editing kind of a special issue of a journal. The 2 co-editors that I'm working with are from the UK and France. So they are and have been collaborators. I've had collaborators in Mexico and South America, the US, Japan.
So is it, do some of these collaborations span long periods of time, or are they sort of project based? You collaborate with this person for that project, collaborate with this person for that project, or is there somebody or people that you have worked with for long periods of time?
I mean, a little bit of both. Often, I mean, especially when you're talking about neuroscience tends to be really kind of technique driven. That often it takes, many years of experience, sometimes, many hundreds of thousands of dollars worth of equipment to really develop expertise in this one thing. And so often you end up, your contribution, once you have this expertise, you often end up contributing to other projects, in part because they have a question that they realize is
they need that technique for.
Yeah, right, is really suited to the technique you have, in your wheelhouse, or vice versa. And so there's a lot of, technique driven dovetailing that happens there. That tends to be, a project at a time.
Right.
Where I have a handful of people that often, friends of mine that I've known for many years. In some cases, these kind of ex postdocs, once they go on to have faculty positions, will continue to collaborate.
Right. Yeah. You said friends that you've known for a long time. One of the things I think about when I think about, well, at least in science, the collaborative model, the sort of party line story of collaboration is they know how to do something you don't know how to do, and together you can answer some question, but I'm often surprised, or not surprised at all, really, I guess. But I often smile because lots of those are also, they're just people you like to work with. They're fun. They're friends of yours. I mean, I've been involved in some collaborations which were not about either of our research. It's just thought it'd be fun to do something together. Let's try to think of something we could do together that we'd both be interested in, kind of thing. And I don't think that human side of the collaboration is sort of well known outside of academia anyway, and maybe not, maybe even outside of our own disciplines. So when you said that, it made me smile.
Yeah, I mean, that's just a natural, at least for my case, I'm sure this is your experience too. That's just a natural extension of, you have these friends that you maybe went to grad school with, postdoc together, and you see them over the course of many years, and you go to conferences together, you're attending the same talks, you're attending the same posters, you go for lunch afterwards, and you talk about something great you saw in a poster, and you say, wouldn't it be wonderful if, and it just can't help but kind of gel into a collaboration.
Yeah. The sad side of that, though, I'm starting to realize now at my academic age, is my longtime collaborators are all retiring now. Oh my God, I'm the only one left. Anyway, that's an aside.
That's why you collaborate with your ex-students and postdocs.
So you can keep doing that. But then you're doing that to them, right? They're on the other side of that. You retire eventually and leave them. Yeah.
Circle of life.
That's right. The circle of academic life. Diano, so if we think about work that you're doing, like really right now kind of thing, like what have you found out recently that's, you know, surprised or maybe excited you, something that you weren't expecting? Do you have any?
Oh, I mean, the bird project that I described to you is very, you know, surprising. Definitely not what we expected to find.
Right.
And it's in many ways, you know, the at the same time, the best and the worst kind of project, because it creates a whole bunch of questions and no answers.
Yeah. I got a lot.
Yeah. So we're left right now, you know, scratching our head about this and saying, okay, something about what we know is wrong, but what is it? And so now it's a matter of, you know, it may very well be a whole other grant cycle to try to answer that question of what don't we have right?
Yeah. So I assume you're going to continue on with that collaboration and that project and following some of those questions through.
Absolutely.
Yeah.
And even that's part of so I mentioned GNOME, but that's part of, you know, a larger collaboration where we have worked with David White, who's also in the psychology department, with people at AFAR. So this is the Advanced Facility for Avian Research. I hope I got that right, at Western.
Okay.
And so there's kind of 3 key faculty members there. So I'm just saying, especially in neuroscience, I think you can't help it because it's so, you know, multidisciplinary. Things tend to balloon into large collaborations very quickly.
Yes. At Western, do you collaborate with David Sherry? Is he one of the
people?
Because I know he's worked on avian spatial memory kinds of stuff for a long time.
Yep. And when I was first, you know, my expertise is in working with mammals.
Yeah.
Right. So that's where I have decades of experience, at least. So working with birds was new to me, and Dave, to his credit, you know, did a lot of mentorship in helping me get my experimentation right. And he's very much, you say, this part of the circle of life that they retire and leave you behind. Dave just recently retired himself. Yeah.
Oh, okay. Very interesting. You know what? In the sort of, I'm not thinking here about specific result or something like that, but in general, what do you think your colleagues can learn from your work? Hard question, I know.
I think for you know, non-neuroscientists, especially maybe non-psychologists who don't think about, you know, the brain quite as much as I do. Yeah. It's really, I often find that people are surprised when I describe how much, you know, a rat or a mouse or, you know, a pigeon can do.
Yeah.
And their abilities. And I think one thing is maybe to, you know, appreciate that. And maybe, you know, maybe not quite as, you know, the things that we can do, a pigeon or a rat maybe can't do quite as fast or quite as often, but usually the differences are in, you know, amounts rather than kind.
Yeah. So I've heard some of these descriptions are often, you know, in that context of what an animal can do is it's often sometimes compared to early stages of human development, right? You say, well, I'm making this up, but like a rat can count as well as a 4-year-old child or something like that. You hear those kinds of comparisons quite regularly, and I think that speaks to your sort of quantity rather than kind of response.
Yeah, absolutely. I think
give me an amazing memory task example from one of your study animals, one of your study systems. What would shock me that they can do?
Okay, you take a rat, for instance, show him an object. This can be anything, a piece of junk. Often we use bits of Lego. Mm-hmm. This is a good example. You take a bit of Lego. It sounds weird as a thing to use for science. But if you talk about doing something like object recognition, Lego are actually great because they're standardized and they're all the same, you know, shape and color. And so, you know, you talk about wanting replication in science. You describe how you put together a Lego object. put a picture of that in a paper, and everyone in the whole world can do it. Right. And most importantly, you can take that thing and describe how you swap around the blocks. And not only do you have two objects that are different, but you can quantify how different they are. So these are some of the ways Lego are great. So keep your kids' Lego. You'll need it. Or send it to me. I'll take it all. So you can take a rat, show him this random collection of Lego, which she has no experience with, has never seen before. Show it to him for 5 minutes. 5 minutes and only 5 minutes. Put him, back to his regular home to merrily live his life for a month. Very long, and in rat years, when you're talking about an animal that only lives for 2 years, a month is a long time.
Yeah.
Put him back into a enclosure, a little testing arena. Give him that same Lego arrangement and a different one. And different, we can mean take the same Lego blocks, right? Exactly the same Lego blocks, put them in a different arrangement. And he can tell you, and rats are very good at telling you because rats love novelty. So if you give them something they've seen many times before and something that's new, they will go and preferentially hang out with the new thing. So it gives you a very easy behavioral test to whether or not a rat recognizes that this is new. So it's a very nice thing you can play with. So you can take this Lego object that a rat has seen for 5 minutes a month ago, and he can differentiate that from a novel arrangement of Lego blocks.
I mean, I'm being anthropomorphic here, but you could say, oh, I've seen that one before. This one looks different, kind of thing.
Right, and he will, preferentially explore that new thing. Honestly, I don't know that I could do that.
I couldn't do that. I could already say that. To me, I have it was yesterday, this week, and the past. That's about all I can differentiate anymore. Diano,
for us, for people, this is fun, a little bit sad, depending on what age you are, but that's something that degrades very quickly with age.
Yeah,
actually, amongst the kinds of memory that are vulnerable to age, and by about 40, you start to see a decline in that the kind of high interference recognition memory that goes into being able to do that.
Yeah, thanks for that cheery thought there, Diano. I appreciate that. Well, folks, we've unfortunately come to the end. I'm sure we could continue this conversation for many hours. Please join me in thanking our guest. Diano, thank you so much for sharing your obviously evident passion for your scholarship with us today. We really appreciate that.